Oh, oh, oh. Oh, oh, oh. Oh, oh, oh. Oh, oh, oh. O'Reilly's. Ladies and gentlemen, ba-da-ba-ba-ba. We're back. Another great night here. Dreamcats Radio. Just a quaint little show in Missoula, Montana. But you might be tuned in Frenchtown, Montana. Or perhaps the Bitterroot Corridor. Or maybe somewhere else on planet Earth. On the internet. 1015kfgm.org. There's a lot of internet out there. It's pretty much wherever you want it to be at this point. But you know what is even more pervasive than the internet on planet Earth? You guessed it, sticks. We're talking sticks tonight on Stick Talk. I don't want to sound drastic, but there's a lot of sticks, a lot of kinds, a lot of shapes and sizes. We love sticks out here. Love just holding a nice stick in my hands, you know? It almost has like a holistic feeling to it, you know? A mystical kind of magic, you know, when you're surrounded by sticks. But you know, I've noticed you know, if you listen to this show often, I don't think you'd pen me as a conspiracy theorist, you know, but I really think there's a place that you don't see sticks a lot, you know. I'm trying to be optimistic here, but stick with me. I just feel like when you're inside, there's not a lot of sticks. Like I'm in the studio here right now, KFGM, at the public library is where I'm broadcasting from. Nary a stick in my line of sight. You know, I don't think that I'm that eccentric to, you know, I'm just pro stick, you know. I don't know when it started, you know, Stick Talk. We've been doing this for years. I come in the library, no sticks. I don't understand it, you know. Outside, I can point to a stick, you know. I get in here, how did we get so anti-stick in this country, you know? I think it would be kind of artistic to let some of those sticks just kind of come on in, you know. So next time you're out and about, just take a couple sticks with you. Just bring them inside, you know. Why do we have to separate ourselves from nature? Is it really that logistic? Is it really realistic to think that we can, you know, separate ourselves from the sticks? I don't like that, you know? We came from nature, you know? Nature is stick. When did we separate ourselves from the sticks, you know? At one point in history, we were sticks. You know, we evolved from the stick. I'm not making this up, folks. But it's pretty cathartic to talk sticks here on Stick Talk. 1015 Stick Talk. You know, but I don't want to be a stickler. I'm not trying to, you know, I'm not trying to be too crazy here. Not trying to poke you or anything with a stick, but I'm just saying, stick with me, you know? You gotta stick it to the man sometimes, as they say. yeah. We're talking about, tonight on Dreamcats, we're talking about one of my favorite species, the tardigrade. Also known as the water bear, or the moss piglet. Johann August Ephraim Goes originally named the tardigrade, meaning little water bear in German. Today, they are often referred to in German as Björnchen, or little bear animal. The name water bear comes from the way they walk, reminiscent of a bear's gait. The name tardigradum means slow walker and was given by Lazzaro Spallanzani in 1777. The largest adults may reach a body length of 1.5 millimeters. The smallest, below 0.1 millimeter. Newly hatched tardigrades may be smaller than 0.05 millimeters. For comparison, grass pollen is typically 0.025 to 0.04 millimeters. Tardigrades are often found on lichens and mosses. For example, by soaking a piece of moss in water. Other environments in which they are found include dunes and coasts, generally soil, leaf litter, and marine or freshwater sediments, where they may occur quite frequently, up to 25,000 animals per liter, or 95,000 animals per gallon. One tardigrade, Echinisocoidus wyethi, may be found on barnacles. With the exception of 62 known species that live exclusively in fresh water, all non-marine tardigrades are found in terrestrial environments. Because the majority of the marine species belongs to Heterotardigrada, the most ancestral class, it confirms the phylum's marine origin. Thank you to Ollie Elms for this atmospheric guitar. Tardigrades have a barrel-shaped body with 4 pairs of stubby legs. Most range from 0.3 to 0.5 millimeters in length, although the largest species may reach 1.2 millimeters. The body consists of a head, 3 body segments, each with a pair of legs, and a caudal segment with a 4th pair of legs. The legs are without joints, while the feet have 4 to 8 claws each. The cuticle contains chitin and protein and is molted periodically. The first 3 pairs of legs are directed downward along the sides and are the primary means of locomotion, while the 4th pair is directed backward on the last segment of the trunk and used primarily for grasping the substrate. Tardigrades lack several Hox genes and a large intermediate region of the body axis. In insects, this corresponds to the entire thorax and the abdomen. Practically the whole body, except for the last pair of legs, is made up of just the segments that are homologous to the head region in arthropods. All adult tardigrades of the same species have the same number of cells. Some species may have as many as 40,000 cells in each adult, while others have far fewer. The body cavity consists of a haematocoel, but the only place where a true coelom can be found is around the gonad. No respiratory organs were found, with gas exchange able to occur across the entirety of the body. Some tardigrades have 3 tubular glands associated with the rectum. These may be excretory organs, similar to the Malpighian tubules of arthropods, although the details remain unclear. Also, nephridia are absent. The tubular mouth is armed with stylets, which are used to pierce the plant cells, algae, or small invertebrates on which the tardigrades feed, releasing the body fluids or cell contents. The mouth opens into a triradiate, muscular, sucking pharynx. The stylites are lost when the animal molts, and a new pair is secreted from a pair of glands that lie on either side of the mouth. The pharynx connects to a short esophagus, and then to an intestine that occupies much of the length of the body, which is the main site of digestion. The intestine opens via a short rectum to an anus, located at the terminal end of the body. Some species only defecate when they molt, leaving the feces behind with the shed cuticle. The tardigrade nervous system consists primarily of the brain and 4 segmental ganglia associated with the 4 body segments. The brain comprises about 1% of the total body volume. The brain develops in a bilaterally symmetric pattern. Tardigrades have a dorsal brain atop a paired ventral nervous system. The brain includes multiple lobes, mostly consisting of 3 bilaterally paired clusters of neurons. The brain is attached to a large ganglion below the esophagus, from which a double ventral nerve cord runs the length of the body. The cord possesses 1 ganglion per segment, each of which produces lateral nerve fibers that run into the limbs. Many species possess a pair of rhabdomeric pigment cup eyes, and numerous sensory bristles are on the head and body. And of course, tardigrades all possess a buccopharyngeal apparatus, a swallowing device made of muscles and spines that activates an inner jaw and begins digestion and movement along the throat and intestine, which, along with the claws, is used to differentiate species. Although some species are parthenogenic, both males and females are usually present. although females are frequently larger and more common. Both sexes have a single gonad located above the intestine. 2 ducts run from the testes in males, opening through a single pore in front of the anus. In contrast, females have a single duct, opening either just above the anus or directly into the rectum, which forms a cloaca. Tardigrades are oviparous, and fertilization is usually external. Mating occurs during the molt, with the eggs being laid inside the shed cuticle of the female and then covered with the sperm. A few species have internal fertilization, with mating occurring before the female fully sheds her cuticle. In most cases, the eggs are left inside the shed cuticle to develop, but some species attach them to a nearby substrate. The eggs hatch after no more than 14 days, with the young already possessing their full complement of adult cells. Growth to adult size occurs by enlargement of the individual cells, rather than by cell division. Tardigrades may molt up to 12 times. Tardigrades tend to court before mating. Courtship is an early step in mating and was first observed in tardigrades in 1895. Research shows that up to 9 males aggregate around a female to mate. Scientists have reported tardigrades in hot springs, in glacial cryokinite holes on top of the Himalayas, and in the deep sea, from the polar regions to the equator, under layers of solid ice and in ocean sediments. Many species can be found in milder environments, such as lakes, ponds, and meadows, while others can be found in stone walls and roofs. Tardigrades are most common in moist environments, but can stay active wherever they can retain at least some moisture. Tardigrades are thought to be able to survive even complete global mass extinction events caused by astrophysical events such as gamma ray bursts or large meteorite impacts. Some of them can withstand extremely cold temperatures, down to 0.01 Kelvin, also known as negative 460 degrees Fahrenheit or negative 270 degrees centigrade Celsius, close to absolute zero. While others can withstand extremely hot temperatures, up to 420 degrees Kelvin, 300 degrees Fahrenheit or 150 degrees Celsius, for several minutes. Pressures about 6 times greater than those found in the deepest ocean trenches. Ionizing radiation at doses hundreds of times higher than the lethal dose for a human and the vacuum of outer space. Tardigrades that live in harsh conditions undergo an annual process of cyclomorphosis, allowing for survival in sub-zero temperatures. They are not considered extremophilic because they are not adapted to exploit these conditions, only to endure them. This means that their chances of dying increase the longer they are exposed to the extreme environments, whereas true extremophiles thrive in physically or geochemically extreme environment that would harm most other organisms. Tardigrades are one of the few groups of species that are capable of suspending their metabolism. While in this state, their metabolism lowers to less than 0.01% of normal, and their water content can drop to 1% of normal, and they can go without food or water for more than 30 years, only to later rehydrate, forage, and reproduce. Many species of tardigrade can survive in a dehydrated state for up to 5 years, or longer in exceptional cases. Depending on the environment, they may enter this state via anhydrobiosis, allowing tardigrades, along with some other micrometazoans, such as worms, rotifers, and crustaceans, protozoans, and plants, the ability to survive in habitats that might otherwise be fatal. In addition to offering protection from desiccation and freezing under normal circumstances, anhydrobiosis also permits resistance to unnatural abiotic extremes, such as sub-zero temperatures, cryobiosis, osmobiosis, or, of course, anoxybiosis. Their ability to remain desiccated for such long periods of time was thought to be dependent on high levels of non-reducing disaccharide trehalose, which is commonly seen in other organisms that survive desiccation. And tardigrades have trehalase genes. However, it has been seen that both in tardigrades and bdelloid rotifers, there is only a partial capability to synthesize trehalose in quantities that may contribute to desiccation tolerance. In response to this finding, more research was done on how these animals survive such extreme conditions. It was found that intrinsically disordered proteins were highly expressed in response to desiccation in tardigrades. Additionally, 3 new IDPs were found to be specific to tardigrades and coined tardigrade-specific proteins, TDPs. These TDPs may maintain the structure of membranes by associating with the polar heads of the phospholipids bilayers, avoiding structural damage upon rehydration. Also, TDPs, being highly hydrophilic, are thought to be involved in a vitrification mechanism, where a glass-like matrix forms within cells to protect the cellular contents upon desiccation. Their DNA is further protected from radiation by a protein called DSUP, DSUP, short for damage suppressor. In this cryptobiotic state, the tardigrade is known as a TUN, T-U-N. Tardigrades can survive and extreme environments that would kill almost any other animal. Extremes at which tardigrades can survive include those of temperature. A tardigrade can survive a few minutes at 304 degrees Fahrenheit, 30 years at -4 Fahrenheit, a few days at -328 Fahrenheit, or even a few minutes at -458 Fahrenheit. Research published in 2020 shows that tardigrades are sensitive to high temperatures. Researchers showed it takes 48 hours at 98 degrees Fahrenheit to kill half of active tardigrades that have not been acclimated to heat. Acclimation boosted the temperature need to kill half of the active tardigrades. Tardigrades in the ton state fared a bit better, tolerating higher temperatures. It took heating to 180 degrees Fahrenheit to kill half of ton state tardigrades within 1 hour. Longer exposure time decreased the temperature needed for lethality though. For 24 hours of exposure, 145 degrees Fahrenheit was enough to kill half of the ton state tardigrades. Tardigrades are the first known animal to survive after exposure to outer space. In September 2007, dehydrated tardigrades were taken into low Earth orbit on the Photon M3 mission, carrying the Biopan astrobiology payload. For 10 days, groups of tardigrades, some of them previously dehydrated, some of them not, were exposed to the hard vacuum of outer space, or vacuum and solar UV radiation. Back on Earth, more than 68% of the subjects protected from solar UV radiation were reanimated within 30 minutes following rehydration. Although subsequent mortality was high, many of these produced viable embryos. In contrast, hydrated samples exposed to the combined effect of vacuum and full solar UV radiation had significantly reduced survival, with only 3 subjects of Millennium tardigradum surviving. Also, it was found that the space vacuum did not have a significant effect on egg laying in either R. conifer or M. tardigradum. However, M. tardigradum, exposed to UV radiation, had a lower egg laying rate. In May 2011, Italian scientists sent tardigrades on board the International Space Station, along with extremophiles on STS-134, the final flight of Space Shuttle Endeavour. Their conclusion was that microgravity and cosmic radiation did not significantly affect survival of tardigrades in flight, and stated that tardigrades represent a useful animal for space research. In November 2011, they were among the organisms to be sent by the US-based Planetary Society on the Russian Phobos Grunt mission's Living Interplanetary Flight Experiment to Phobos. However, the launch failed. In August 2019, scientists report that a capsule containing tardigrades in a cryptobiotic state may have survived for a while on the moon after the April 2019 crash landing of Bereshif, a failed Israeli lunar lander. But in May 2021, it was reported that they were unlikely to have survived the impact. In recent years, there has also been increased speculation regarding tardigrades' ability to survive on Mars without any life support systems. But it would still need stuff to eat to survive. Thank you, my dear listeners, for joining me on Dreamcats Radio. In this dimension, I broadcast from 10 to at least midnight, every Sunday night, on Missoula Community Radio.org. Sweet dreams.